Hi guys, welcome to your cell cycle notes for this unit. So we're going to first start by talking about just cells in general. So should they divide or do they not divide? And how do you grow? Well, as you're developing, even way back to when you were just a zygote, you grow by both more cells and bigger cells. But cells don't just keep growing infinitely. If they get too big, they can't do their processes and carry things out efficiently. So there's a couple things that limit cell size. Things like surface to volume ratio and their metabolic requirements. So how much do they truly need to function? So the major question then becomes, how do cells get bigger? And they do this by dividing. So before we get into the actual division, let's talk about kind of the brains of the cell and what is kind of regulating that division. So hopefully you've heard of what a chromosome is before. So a chromosome is just a coiled piece of DNA that contains genes. There's a couple very similar sounding words that you need to be familiar with. So first of all, a chromosome is an X, right? But there's two parts that come together to make that X. So each of those parts is a sister chromatid. They connect by something called a centromere. Lots of C words, right? But one that isn't a C word is the telomere. And this is kind of the ends of each of these arms. I'm just going to label it with a T. So there are 4 telomeres on a chromosome. Like I mentioned, it's a coiled piece of DNA. So this image just breaks it down a little bit more for you. So you have your DNA double helix here. It's going to coil around these histone proteins, which then form something called nucleosomes, which then further coil to make the X shape of your chromosome, which is then located in the nucleus of the cell. All right. Now it's time to get into the fun part, the cell cycle. So mistakenly, we tend to think of just mitosis when we think about the cell cycle. And in reality, if you look at this picture over here, mitosis is only a very small part of the cell cycle. Interphase makes up the majority of it. So we're first going to start talking about interphase, which is, like I just said, where cells spend most of their time. Then we will break down the 4 steps of mitosis, followed by cytokinesis, which is that final division of the cells. All right, so interphase. This is where the cell is preparing for division. So it's not dividing here yet, it's just preparing. And to prepare, there's 3 different stages that the cell has to go through. And some really important things happen at each stage. All right, so first stage is G1. So G1 is also known as the first gap, and this is where the cell just simply grows and it creates more organelles. So if a cell is going to divide in half, it can't have half the number of mitochondria, half the number of Golgi, half the number of ribosomes, because then it won't be functioning at an optimal level. So it needs to create more organelles first. Now, super important, and there's one of these at every stage in interphase, there is a checkpoint. So cells are not allowed to grow unless they're given a specific signal. So if they're not given the signal, they enter something called G0, which is like cell rest or cell timeout, however you want to phrase it. But in G0, there's just that. There is 0 growth. And actually, a lot of your cells stay here normally. So things like your nerve cells, your muscle cells stay in G1 or G0, excuse me. All right, now we go to S. So S for synthesis or replication of DNA. So this is where those chromosomes get duplicated because once again, mitosis, we're starting with a diploid cell. So say if it's a human cell, you're starting with 46 chromosomes. And then when you end, you're ending with 2 cells, each with 46 chromosomes. They don't have half the number. So it's important that DNA is first replicated before it divides. And then lastly is G2, which is known as the second gap. And once again, this is just kind of those final checkpoints, kind of like before you would take off on a plane, that all systems go, like make sure everything is functioning properly in order to divide, because the body doesn't want to divide bad cells. So this is that final checkpoint before they're allowed to go into mitosis to divide. All right, mitosis. So mitosis is probably something that you're very familiar with. This picture you see is a bunch of different plant cells in various stages of mitosis, and we'll do some activities with that in class so you can see that better. But let's talk about these stages. So there's 4 stages, P, M, A, T. So PMAT, so Prophase, Metaphase, Anaphase, Telophase. Let's get into them. Prophase. So prophase is the longest phase of mitosis. And there's a reason for that, because a lot of things happen here. So the chromatin has to coil into chromosomes. That's super important, right? The nucleus has to disappear. That way the cell can divide and split those chromosomes. And also something called centrioles. They look like little pasta noodles. Those form at opposite ends because the spindle fibers are going to grow out of those. And the spindle fibers are ultimately what helps this process happen. Metaphase, think middle, right? Meta, middle. So in metaphase, all of the chromosomes line up in the middle, single file, okay? Because every single one of them has to split in half. So if 2 are next to each other, that's not going to be very helpful, right? So they line up single file. The spindle fibers, they come all the way across and they attach to, there's that word again, this centromere or the middle of the chromosome. Next we have anaphase. So in anaphase, those spindle fibers, they're all the way out, they're starting to pull back, they're starting to shorten. And as they shorten, they were already attached to the chromosomes at those centromeres, so the chromosomes split. So they split into 2. And those move to opposite ends of the cell. And these new chromosome halves are something called chromatids. All right, we're almost done dividing. Now we have telophase. So in telophase, they're fully separated. The chromatins are going to start unraveling back into chromatin because that's kind of their natural form of existence in the body. The spindle fibers break down because, well, they don't have a role anymore because they've already pulled the chromatids to the opposite sides, and the nucleus begins to reform. The plasma membrane also begins to pinch, but this is super important. It begins. A lot of people think telophase is where that final division occurs, but it's not. So telophase is a pinching in, and then in cytokinesis, this is where we get that full division. So we get 2 separate nuclei, 2 separate cells, the cell membrane fully pinches off, and you get 2 daughter cells. But there's more, because we were just looking, all these pictures, right, are all animal cells. Well, plant cells are a little different because they have a cell wall. So plant cells can't really pinch in like an animal cell would. So there's a different terminology depending on the type of cell. So if you are an animal cell, you form something called a cleavage furrow as you're dividing. And if you're a plant cell, you're going to form a cell plate as you divide during mitosis. All right, so we've talked about in general how cells divide, but there's also a little bit of differentiation, right? Because not every cell in your body is exactly the same and does the same thing. So genes are going to tell the cells what to do. And depending on if these genes are on or off, depends on what type of cell it is. So it's kind of like blueprints, right? So if you live in a neighborhood, chances are every single house in the neighborhood is not 100% identical. They're a little bit different in their own way based on those blueprints. So cell division is it's a very, very controlled process by the body. And it's controlled mostly within the cell, but outside influences can factor too. So things like growth hormones, for example, can help stimulate cell division. But now the bad part, right? What if cell division isn't controlled? Well, unfortunately, that typically is what we would call cancer. So cancer is uncontrolled cell division of abnormal cells. So normal cells aren't going to have uncontrolled division because they are regulated. So abnormal cells can do uncontrolled division and they can fly past those checkpoints in interphase because they are abnormal. They have a mutation, they have something wrong with them where they're not going to follow through and they kind of override the body. So the reason why cancer then typically kills is because those abnormal cells that are just dividing are ultimately going to crowd out the normal cells. So then we also have our two types of tumors. And you may have heard of these two types before. So a benign tumor is kind of the better tumor to have if you're going to have a tumor because it's contained. So it's usually in a specific location within the body. A malignant tumor is super bad. You don't want a malignant tumor because that is not contained. It just kind of spreads all over the body. We also then need to talk about treatment. So in terms of how you treat cancer, I'm sure you've all heard of chemo or radiation, right? Well, chemo is a little more severe than radiation because it's going to kill every single cell dividing. And guess what? You have lots of cells in your body that are normal that need to divide. So a lot of times it affects you in more ways than just those cancerous cells. Radiation kills the cancer cells because it's targeted, and it also will kill some of the normal surrounding cells, but those will typically grow back via mitosis. One other type of cell control you need to be aware of is something called apoptosis. So this is actually programmed cell death. So this can be from an internal signal, which is usually the case. Usually the body gives this a signal like this is not a good cell, it needs to die. But it can also be an extracellular signal as well from like a toxin exposure or like too many growth hormones, things like that. So cells though can also just kill themselves off. So it's almost called cell suicide. But I know what you're thinking. Oh my gosh, this all sounds so terrible, right? But it can actually be good too. So let's talk about good versus bad. So apoptosis in a good way is why you have fingers and toes, right? Do you know anybody that maybe has a webbed toe or a webbed finger? My cousin, he has webbed toes, right? So that means apoptosis didn't happen when he was in the womb. So it resulted in those webs. Now, it's also, ladies, in menstruation. So when your uterus sheds once a month, that is a good effective apoptosis because your body is ridding it of those cells. Now, bad effects, they divide slower than they die. So you have cell loss because you have lots of cells dying and they can't recover. But also they can divide faster than they die, which is when a tumor can form. And then lastly, I want to touch on macromolecules, right? So we've been revisiting macromolecules throughout the year so far. And it's important that you know that proteins are very important in mitosis. So these things called cyclin proteins are going to bind to these enzymes, cyclin-dependent kinases, CDKs. Remember, ASE is an enzyme or a protein. And what they do is they control the stages from starting the next process. So they're really, really important in those checkpoints. They're important to signal the DNA to replicate. They're important to signal the cell to start dividing. They're important to signal like starting a new cell cycle. So these kinases are super important. And remember, they're proteins because they end in ACE. All right, and that is it for your notes.